Hi, this is Miss Litton, and these are some lovely AP biologists coming in at lunch to review Chapter 7, Photosynthesis. So, in general, moving through here rapidly, we know that plants are what? What could we categorize them as?
Eukaryotic.
Okay, they are eukaryotic, but I said plants, so they are what?
Autotrophs.
Autotrophs, good, or producers, and we are what?
Heterotrophs.
Heterotrophs. So when we think about the reactants, I would, though I don't have it on the board right now, I would be able to write the equation for photosynthesis without any problem. I would be able to identify what's getting reduced and what's getting oxidized in the process. And I would be able to identify where the reactants are coming from. So for instance, tell me the equation for photosynthesis. What is it?
H12O6.
No.
6 CO2.
Okay, 6 CO2. Keep going.
Plus energy.
H2O.
Okay, plus energy. Okay, keep going.
H2O.
Plus H2O. Good. Goes to what?
C6H12O6.
I don't know what I'm writing, but good. C6H12O6 plus what? Plus oxygen. Okay. So where is the carbon dioxide coming from? Where is the carbon dioxide coming from?
No, us.
It's not coming from the plants. I mean, it could, but most of it's coming from the air, right? Not from the sun. Okay. The CO2 is coming from the air. How is it getting in the plants? Through the stomata, through the leaves, remember? The little openings in the leaves. So the CO2 is going in. Where are the plants getting the energy from?
The sun.
The sun. Okay, so energy is coming from the sun. And here comes the sun traveling in. Particles of light or cold? Good. Where's the water coming from?
Roots.
Roots. Okay, so water is coming from the roots. Okay, the products of that, you can form sugars and oxygen gas, which is then going to either be used when the plants do cellular respiration or released from the plant. Okay, now I want you to think about something for a minute. You are able to tell me great details about photosynthesis, but the very simple question of writing the equation and telling me where everything comes from, that was actually difficult. Okay, so make sure that you're remembering the big ticket items, like where it occurs and how the reactants get in there. Okay? And make sure you can also tell me what's happening to the CO2. Is it getting reduced or is it getting oxidized? Yeah, the CO2 is getting reduced into glucose, right? And, oh my goodness, getting reduced into glucose, and the water is getting what? Oxidized. So let me get a better diagram of that. Okay, so the CO2 is getting reduced. Okay, and the water is getting oxidized. Okay, during this process, we use an energy transfer molecule. What is the name of that molecule? We temporarily reduce it, and then we NADP. Good. So we use NADP. Okay. All right, good. Structure of the plant. We looked at this, right? And remember, this was like your standard one, but do you remember the adaptation that we saw? Yeah, that wreath, that Krantz anatomy. Okay, good. I just want to remind you of that. You have no trouble writing the equation, hopefully out. We talked about, kind of in a summary, the light reactions are going to occur where? Where are the light reactions specifically occurring? Yes, in the leaves, in the chloroplasts, in the thylakoid membrane. So this is part of like that, remember the green hollow pancakes we were looking at? Okay, so when we look at those grana inside the chloroplasts, the light reaction is occurring on them. Okay, and water, what's the point of water? Why are we using water in the light reaction? What is it providing?
It provides energy.
The water doesn't provide energy. The water provides electrons. Good. So where do we get the electrons from?
Hydrogen.
From the hydrogen. So the hydrogen are going to donate their electrons, and their electrons are going to get what? Excited. And when they calm back down, when those electrons calm back down, we can use the hydrogens that donated those electrons to create a chemiosmotic gradient. We're going to concentrate the hydrogen ions to the interior of the thylakoid membranes, interior of those hollow pancakes, right? And we can use that to generate what? ATP. And we're going to get the electrons excited a second time, but this time we're going to give those electrons to NADP, forming reduced NADP. So both of these are products from the light reaction that we will use in the light independent reaction. What's another name for it? The Calvin cycle. And then what's the old school name? Dark reaction. Matt, do you need me, sweetheart?
No.
Okay. And now the CO2 comes into play, and CO2 gets reduced by the electrons that ultimately came from water. And we're reducing it into sugar. Right? And to do that, NADP that was reduced becomes oxidized, so it can be used again. Okay, and ATP becomes ADP, which gets reused in the light reaction. So the light reaction provides for all of the reactants except CO2, right, to do the Calvin cycle. Okay, big ticket item questions, concerns. Okay, can the person whoever's sitting in the blue seat right now just link the light reaction and the Calvin? Oh, by the way, where does the Calvin cycle occur? Stroma. Okay, so just try to put it in your own words right now. Link these two reactions. Go. Okay, that's all the time I'm giving you. So the light reaction requires light, and obviously, and we talked about the absorption spectrum and the ability to harvest that light energy, and different pigments capture different wavelengths of light in their photosystems, which eventually is going to get passed to a reaction center, which is going to then trigger that electron transport chain. We mentioned how you could use a spectrophotometer to measure, okay, that action spectrum. And here's a question. We are 818 number.
I just sent it.
Okay, did we get a hundy? Ah, yes, it does require light, but it also requires membranes. What are the names of those membranes? Thylakoid membranes. And it requires water. Why? Water supplies the electrons and the hydrogens to create the chemiosmotic gradient. Okay? Shadowland, sunshine, you understand? Yes.
So within the water, the oxygen is giving the energy?
Is the oxygen giving any energy? No. The oxygen is a product of splitting the water.
Okay.
What's giving the energy in photosynthesis?
The bonds?
No. The
Oh, okay.
sun.
Right.
The sun. Okay? So when you have a water molecule, it's going to get split. Oxygen is going to be a byproduct. Are you looking up here, Max?
Yes.
Oxygen is going to be a byproduct that just gets released.
Okay.
Okay? Then you have the hydrogen ions, and you have the electrons it contains. The hydrogen ions are going to go be part of your chemiosmotic gradient inside your thylakoid membranes. You're going to concentrate hydrogen ions, yes?
Okay.
And on these thylakoid membranes, you're going to do a na-na-na-na-na-na and a na-na-na-na-na and end up giving these electrons that you excite to NADP, forming NADP, right, reduced NADP, okay? Then you will, by concentrating all of these hydrogen ions inside the thylakoid membranes, when they go back out, you're able to take ADP and convert it to what? ATP. And then you will take this ATP and the reduced NADP to the dark reaction, or the Calvin cycle, or the light independent reaction. Yes?
Is NADPH the same thing as?
Yes, reduced. Sometimes they write it NADPH, sometimes NADPH2, or just NADP with a little r, little e. Okay, good?
Yes.
All right. Do you want to heat that up?
Huh?
Do you want to heat that up?
No, I like it cold.
Oh, okay.
That's weird. Do you heat yours? No, I wish I could have.
You could have. My microwave's
Well,
right.
it's in my belly now. It's not.
Oh, okay. Yes, the primary pigment is chlorophyll. There are carotenoids, there are xanthophylls, but the primary pigment is chlorophyll. Good job, Spanish is so hard.
Yeah, it's not loading.
It will, it will.
Why is it down? Oh, there it is. Uggs. Uggs for the win. You know Uggs are originally made from
Yeah, I pressed the second button down. I thought it was green. I guess it's this one.
I'll drink it. You just take it. Not me. Uggs.
Okay, so when plants are green, okay, I'm got a lot of green on, okay? If you're seeing green, that means the pigments in my dress are not absorbing green. What are they doing to the green? Reflecting it, okay? And the areas that are blue, they are absorbing the green and they're reflecting the blue. So what color does the pigment chlorophyll not absorb since it's green, much like the 91% who got this right? Okay. Oh, hanging their heads in shame. Yeah. All right. Enough said.
Roll on the green. I'm so
Yay! First empty.
Number 5.
Nice.
Frappuccino. Uggs right there.
Good job, hashtag artsy. All right. So, as you can see, and as we've gone over several times here, the light reaction is occurring on these thylakoid membranes, generating ATP, reduced NADP. So out in the stroma, you can do the Calvin cycle. And the Calvin cycle is going to reduce CO2 into a carbohydrate. Yes?
2 questions. One, if the thylakoid membranes are so important, how come they're so small in the cell?
Because think about why are cells small? Increases what? Surface area. Yeah, so having the more membranes you have, remember these are just little proteins embedded in the membrane. So you can be very tiny, and the more of those surfaces you have, the more area you have to do the light reaction. Does that make sense?
Yeah.
You got another question?
Yeah. I'm not sure if like you're going into more detail about the Calvin cycle, but I don't get the step where the
Yes, we're not on that yet,
Okay.
but we will. Okay, so now the light reaction, when we look at that, okay, we are taking water, okay, we are releasing oxygen from that water, taking the electrons. Those electrons are getting excited by what? The sun, okay? Now, the hydrogens from that water, what did we do with those hydrogens? We sweep, we kept them inside the grana, inside the thylakoid membranes, right?
Yeah.
Okay, then they got excited by the sun, then we na-na-na-na-na-na-na-na, and when we na-na-na, we're gonna take PQ, and we're gonna bring even more hydrogen ions to the interior of those thylakoid membranes. And then we can use all of those hydrogen ions when they go back out, then you can make some what? ATP. We're going to take these same electrons that we just used and we're going to excite them again. But this time we're going to give them to NADP to form reduced NADP. This is called non-cyclic photophosphorylation. Non-cyclic because electrons come in from water and go out on the NADP. Photo because it requires what? Photo. Yeah, the sun's light. Phosphorylation, we are making what? ATP. Making ATP. Okay, whereas cyclic, which is not as efficient, those electrons get excited and na-na-na-na-na makes some ATP. Excited, na-na-na-na makes some ATP. Okay, so that's the difference between those two. If we kind of follow it here along the membrane, here you can see the water gets split, sweep, we have our hydrogen ions, our electrons are getting excited in photosystem 2, na-na-na-na, here comes PQ, bringing even more hydrogen ions to the interior, get them excited again in photosystem 1, given to NADP to form reduced NADP. Then all of the hydrogen ions here in the interior are going to come back out, and you're going to synthesize ATP. Yes?
So for cyclic,
Yes.
does that use photosystem color?
Yes, parts of it. It's kind of a folding back of one and two. Yeah. Okay, are you guys all right? All right. So again, putting them together, you can see the electrons that came in water get given to NADP, which get given to CO2, which you use to build your sugar. But you also need the process of, you need the ATP in order to make that reaction go as well. Okay, I have explained the same connection, I believe, 4 different times now, right? Just in this little period. So if you are struggling with that, I'm going to encourage you, there are so many videos associated with your book, with your book's website, okay? Take advantage of those. Just Googling, you will find so many on YouTube. Photosynthesis review, crash course, if you just need to see it one more time, okay? Here you go. Your Bozeman videos, you should be watching those. He does a great job of that. Yay, 2. All right, now, let's move on. Okay, so now we're going to do the light independent reaction, and there are 3 parts to it. You got to fix the CO2, taking it out of the air, okay, making as a solid. You are going to do CO2 reduction using the reduced NADP as a source of those reducing electrons, and you're going to use some ATP. At this point, you've really achieved your goal. But if you stop right now, you can't keep the party going. So that's why we only skim 1 off the top and we leave the remaining 5 to go through the cycle again. So that's why we need to do the 3rd part, which is the regeneration of the RUBP. Okay, and then I talked to you using this one. And if you weren't in my 3rd period class yesterday, we recorded and put on YouTube just me doing this with this one, if that is helpful to you. Okay, so you have five carbons in each RUBP, and each one is going to hook up with the CO2, and 5 plus 1 is what?
6.
6. Is this a stable molecule? Is it? Yes or no? This 6 carbon molecule, is it stable? No. So what is it going to do? Break in half. What was the name of the enzyme that hooked the CO2 onto the RUBP?
APBio.
Yeah. RUBP carboxylase, or another name for it, which is what you were trying to say, Rubisco.
Yeah.
Okay. So now I have 3 6-carbon units. Each one of those is going to break in half. Break in half. So I have 1, 2, 3, 4, 5, 6. But now I need to level them up. So for all 6 of those are going to receive an ATP. All 6 are going to be reduced by NADP. And now I have 6 PGAL. 6. Now each of those represents what? Guys, that's super distracting to me. I can't even begin to tell you. Each of those is going to represent what? What is a PGAL or a G3P? A half of a glucose. Okay? Each one of these is a half of a glucose, and another name for this is G3P. Okay? It's a half of a glucose. Now, I take one away, that leaves how many left? 5. If you use some more ATP, you could take 5 3-carbons and change them back into 3 5-carbons. Do this whole thing all over again, take another one off, and now I have 2 halves of glucose, which means I have 1 whole glucose. Okay? So I bring 3 CO2 in at a time. So CO2, CO2, CO2. Each CO2 has how many carbons in it? 1, right? Each carbon dioxide has 1 carbon. So carbon, 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 3 carbons in, 3 carbons out is a half of glucose. Again, I bring another 3 carbons in and 3 carbons out as a half of a glucose. Does that make sense?
How come you can't just add more carbon at the top and then take more out when you're like taking it away?
You're gonna need help me on that. This cycle is happening all the time.
Yeah.
It's continuously. It's not stopping as long as the light reaction is providing the ATP and the reduced NADP.
Yeah, I understand that part, but like, how come you can't just take a full glucose out? It's only half.
Oh, okay. Because then you wouldn't have any more RUBP. You would have depleted it. See, this site, it's coming pumped and loaded with already 15 carbons, right? So you're adding 3 in and using that as a base in order to create 1 half of a glucose, and then you keep that going. You keep reinvesting your carbons. Okay? All right. Let's do, and then we talked about, we didn't get a chance to do the songs through, but the songs are recorded for you, okay? But that could help you with that. But let's do the questions we did not get to yesterday, because I just did all yeah. Okay, last but before I leave is RUBP. Sometimes it's naughty and binds with CO2 instead of O2. Now you have to know this for me because it's in our notes. I just told you photorespiration and the different mechanisms to deal with that is not on the AP exam, but you have to know it for me because it is in our notes. And we talked about the 2 strategies. One was the C4 plants, where they isolate the Calvin cycle into the bundle sheath cells. And the second example I gave you were the CAM plants, where they isolate they take the CO2 and fix it at night when it's safe, so during the daytime when they're doing photosynthesis, CO2 can be re-released. Okay. So which of these statements is true? That means all the rest of them are what? False. What's your answer?
Yeah.
A. Yeah. Okay, it is not the only pigment in the leaves. Okay, there's xanthophylls, there's carotenoids. Those are ones as well. We already did that one.
Wait, that was A. I got 10.
To make some. Okay. Now that you just said
But
it.
yeah,
You're like, I am singing.
Dude, I went to
Okay, yeah, to make some ATP.
Do you know what the map is? Over there.
Okay, a substance that, oh, did we do that? No. A substance that reflects or absorbs a specific wavelength of visible light is a pigment. And we already did that one. Complete this equation.
6 from there.
What's your answer there? Oxygen, right? Oxygen. You can give me just a second. I'm doing a review. Okay. We did that one.
Stop.
Solar energy is absorbed and water is split during the what? Light dependent reaction. Light dependent reaction. We already did that one, and that was the end. Okay, review. And review your glycolysis too. That was already up there. Okay, have good days.